Hello, I'm Simona Colla, and I'm an associate professor in the Department of Leukemia at MD Anderson Cancer Center. My laboratory at MD Anderson is currently working in trying to understand the biology, the molecular mechanism through which a cell from MDS expand into the bone marrow in the patient, with the final goal to find a new therapeutic approach for this disease that so far remain incurable. The lab is working on multiple different things and approaches, mainly thanks to the support of the Moonshot program at MD Anderson. We are interested in trying to develop a new therapeutic opportunity to overcome anemia in a patient with myelodysplastic syndrome. This is current our moonshot proposal. We are currently working with Ajax to develop a small molecule inhibitor of a specific pathway named HRI that we think is fundamental to drive aberrant erythroid differentiation, and so for this it results in anemia in the patient, and anemia remains one of the most important phenotype in this patient, because although patients do not really die of disease by itself, the need of transfusion can induce a huge toxicity in the blood of this patient, and this patient develop a problem in liver, in the heart, and so at the end can have organ failure. So one of the projects in the lab is really to try to collaborate with Ajax to develop this small molecule to target this pathway, and now this is seen to work not just for myelodysplastic syndrome, but we have also extending the approach to patients with a sickle cell disorder. Other projects in the laboratory, and this is mainly what I presented here today in my talk, are trying to prevent the development of MDS by targeting the early stage of the disease that is named clonal cytopenias of undetermined significance. Is an intermediate phase of the disease, not MDS, but before, when only a minimal proportion of the cell into the bone marrow are mutated and the symptoms in patients are limited. So it's easier potentially to target the leukemic clone here to allow the normal expansion of other wild-type cells into the bone. And to do so, we are using multiple approaches. One of these is a collaborative effort with the Department of Stem Cell Transplantation, in particular with the group of May Daher and Dr. Katie Resvani. We are trying to develop an adaptive NK strategies in a way that we are able to inject into patient NK cell from a healthy cordon blood donor, and we are modeling this system inside the mice right now. And the reason is because what we observed was as an initiation event, this SICA cell, this early stage cell, are able to escape the immune surveillance by aberrant function of this NK cell population. And this was part of the talk today. Moreover, what we are trying to develop is a new therapeutic approaches to overcome failure to hypomethylating agent. So MDS, the standard of care in MDS remain after years and years the therapy with hypomethylating agent. However, 50% of patients only respond to this therapy, and other 50% of the responder can develop disease progression and progress to acute myeloid leukemia, and at this stage the disorder become completely dismal and poor, and patient can survive only for 4-6 months. So here, during the last 5 years, our lab has published an important manuscript in which we dissected the stem cell architecture in MDS, and we found that only half of patients at HMA failure can indeed respond to the new therapy with BCL2 inhibitor venetoclax. This is a completely novel therapy in MDS, very promising, but not all patients responded. And what we found that indeed is the stem cell architecture that drive the response, because only 50% of the patients become dependent on BCL2 at the time of progression. However, we also found that in other 50% of the patients, their response can occur anyway later, although the stem cells are not depleted. And so we were trying to investigate why this happened. And we discovered that in this patient there is a particular subpopulation of T cell, CD4 T cell, that is increasing at the time of marrow complete remission and decrease again at the time of progression. And now the lab is focusing in trying to understand the function of this particular subtype of CD4 cell that we identify as a stem cell memory CD4 T cell. This T cell has shown by single cell RNA sequencing analysis a signature of cytotoxicity and effector function, and so seem able to directly kill the blast, the tumoral cell, into the patient. So now we are modeling in vitro using functional assay their function. And we have now a collection of 64 samples that we are analyzing from patients enrolled in clinical trial of venetoclax who has progressed after HMA failure. So basically, this is the major program that we have in the laboratory. The laboratory is also working in collaboration with Dr. Neil Young at NIH to try to understand why patients with telomere shortening, so patients with mutation, germline mutation in telomere maintenance gene, have the propensity, the predisposition to develop a myelodysplastic syndrome. And we found that in this patient acquired a specific set of mutation. Among them, the most frequent is a mutation in U2F1S34. And in the lab, we investigated the molecular mechanism through which this mutation induces MDS, and it seemed like that the mutation is able to shut down completely the pathway through p53 activation, and so allowed the clone to expand and overcome the negative effect of telomere shortening inside the tumor.